Okay, I think we're going to go ahead and start now. If you could all make sure that mute is on, so I don't have the noise going on in my earbuds. So if you're not sure where that is, if you take your cursor down to the bottom of the screen and you swirl it around, what you'll see is something that looks like a microphone. If it's white, you're unmuted. If it's red, you're muted. So just go ahead and hit it if it's white and mute yourself. It'll say mute me. And that way we'll be able to
Hit it. You have to hit it.
Oh, no, I'm open.
and then Randy can go ahead and identify people and help them out if we need to. So I just going to start now, just to give an explanation of why I'm giving this lecture. I gave a similar lecture a couple of weeks ago because I stayed connected with most of my friends through Facebook during social distancing and quarantine. And I noticed there was a lot of comments that were coming up and facts that people were spreading on the internet that just simply weren't true about this virus. And so I gave my first lecture about it a couple of weeks ago, and people really enjoyed it. And then I talked to the folks at the University of Akron, and we thought that maybe this is something we could do for the university community and actually the community of Akron as a whole. So as a resource, I'm a microbiology professor at the University of Akron in the Department of Biology. I specialize in microbiology in cave environments, but my PhD is actually in medical microbiology and immunology. I've served on the Professional and Scientific Affairs Board of the American Society for Microbiology for 6 years, and I actually worked with Anthony Fauci in that position. So if Joseph could not share his screen, that would be great. And so I thought, well, you know, I've been teaching medical microbiology for 20 years, and I've been teaching about SARS for over 15 years and MERS for about another 7. So I thought what I could do is take some of the material that I give lectures on at the university and maybe give a presentation for the community. And Randy is the host, and what Randy's going to be doing is keeping an eye on questions. So if you look to the right side of your screen, there is a chat, and it says chat to everyone or chat to Randy. And if you type in any questions there, what Randy can do is curate those questions. And at the end, what we'll do is just start answering those. So I'm going to try and make this a relatively short lecture so that there's plenty of time for questions. I'm hoping that most of what I do will answer people's questions in advance, but please feel free as questions arise to type them in. Randy can then summarize them at the end and we'll go through them. But this is really a chance for you guys to ask me questions about the virus. So the first thing is what is a virus? And the answer is that it is, so this is the technical term that we teach in class. So a mobile particle of genetic material that overtakes a target cell, turning it into a factory to make copies of itself. So it sounds very jargony. So a lot of the words are important in the way that we describe them, because if we say something differently, it doesn't mean the same thing. So genetic material, that's basically the barcode for all life that we use, the information that is inside ourselves to make copies of ourselves. Viruses are not alive, so they're unlike any other microorganisms in that they aren't living. They are only active when they're inside of a cell and growing. So outside of the cell, they're just a particle of genetic material that's wrapped up. And you may have heard it's been in the press about DNA and RNA, and I'll explain a little bit about what those are. But viruses are either a piece of DNA or an RNA packaged inside a protein. And they have a receptor on the outside of the cell that targets them to a cell. They get inside, and then they hijack the host machinery, and then they force that cell to make copies of themselves. So all the normal cellular processes that would be going on get turned off, and then the cell just turns into a factory to make massive numbers of the virus. So the virus isn't really alive when it gets inside the cell. It's just causing the cell to make more copies of itself. A lot of people don't know where viruses come from, and that includes a lot of biologists. They don't have an evolutionary history the same way that animals and plants do. And because of that, it's hard to trace them back to their origins. Where we think they did come from is that when our cells divide, they actually use proteins to make copies of themselves. So one cell will cut its nucleus into two, and then the cell will kind of pull itself apart with a new nucleus going into each copy of the cell. And they do this through a cell cycle. And there's lots of proteins that regulate that. And what we think has happened is that a few of the proteins that control that process kind of got spun out in their own particle with their own piece of genetic material, and then they became self-replicating. So that's where we think viruses came from. So because of that, they don't have a common ancestor. If you can think of them as mistakes in the code, and mistakes arose in the code every now and again, you can see that they just get spun out at lots of different times. And because of that, we don't have a common ancestor. So we can't trace them back the same way you can trace primates back. You can't trace viruses back to an origin. So what we do is we classify them based on their structure. And the structure basically comes down to the kind of genetic material they're made from. They're either going to be made from DNA, which is what we have in our nucleus, or they're going to be from RNA. And the way our cells work is that DNA, which is the code of life, is turned into basically a sheet of paper with instructions on it, and that is the RNA. And then this sheet of paper leaves the nucleus and goes into the cell, where the cell machinery turns it into a protein. So viruses, as I said, were just a piece of genetic material. They're either going to be a piece of DNA or they're going to be a piece of RNA. The coronavirus is an RNA virus, and you may have heard of this already. So it has all the instructions to make proteins. And what happens is when it gets into our cells, what it does is it makes proteins to make copies of itself. So this is kind of like a tree of viruses, but don't think about it as the viruses having a common origin. This is just how we classify them. So this is just the group of viruses that are RNA viruses. The DNA virus group is completely different, things like herpes viruses. Here we look at whether they have basically icosahedral. So do they have kind of a star shape, or are they kind of wound up like a helix? So we divide them into those two groups. And then the other thing we do is we look at whether they have an envelope or not. This group here is the enveloped group. And this is related to how viruses make copies of themselves. If a virus makes so many copies of itself inside a cell that it bursts the cell open, it's kind of sprayed out into the environment. And if that happens, we call it naked virus because it's not covered in anything. However, if the virus buds away from the cell membrane, kind of like a soap bubble, then what it does is it takes a little bit of the membrane of your cell with it. So if you can imagine that this is the membrane of your cell, the virus is coming up to that membrane and it starts to bud out. And what it does is it takes a piece of that membrane with it, and you end up with a virus with a membrane around it. And the technical term for that is enveloped. And that's actually really, really valuable for us because anything that has an envelope is basically surrounded in a cell membrane, just like a soap bubble. And you can imagine how delicate soap bubbles are. So that really helps us decontaminate and get rid of this virus in the environment. If you had something like a picornovirus or a neovirus, something that doesn't have an envelope around it, it can survive on surfaces for months. You may have heard of noroviruses, which cause gastrointestinal illnesses on ships and ocean liners. Those things can stick around for months. The nice thing about a coronavirus is that because it's got that envelope, it has a relatively short period of time that it can survive. So our virus here is the coronavirus. So it's this one right here. So it's a RNA virus. It's helical. You can see it's kind of wound up and it's enveloped. The other ones in this group are rabies and influenza, paramyxoviruses. HIV is right here. This is an unusual run because of the way it makes RNA into DNA in our cells. It doesn't mean that there's any relationship between these viruses and how they cause disease. It just means that based on their structure, they look the same. All right. So that bubble has a big impact on survival. You've probably all heard about cleaning surfaces and touching surfaces and washing your hands. If you have that bubble around you, your membrane, you can survive on a soft surface such as cardboard only for about a day. So if you've got an Amazon package that's being delivered and it takes 3 days to arrive, it's probably a good chance that it's safe. I've noticed that the UPS guy comes to our house and he has gloves on. Hard surfaces are going to be about 2 to 3 days. So that means any kind of hard surface. Most people catch the flu, which is structurally very similar to a coronavirus. They catch the flu from touching door handles. And they touch the door handle and then they take their hand and they put their hand on their face. We know that these viruses get in through what we call mucous membranes. So that's the soft bits. So if you imagine where the soft bits are on your face, it's going to be inside your mouth, it's going to be inside your nose, and it's also going to be inside your eyes. So the soft material around the edge of your eyes is also a mucous membrane. So while you might think that if I breathe in, I might be breathing in the virus. If you touch a door handle and the door handle has virus on it, and then maybe you touch your mouth or you touch your nose or you even touch your eyes, then you can introduce the virus that way. So that's why we talk about hand washing. It's perfectly fine to touch these surfaces as long as you wash your hands before you touch your face. So this is why there's a big emphasis on it. I actually have a pair of gloves that I have in my car. And when I get into my car, I put my gloves on, I go to the post office or I go to the store to shop wearing my gloves. I come out of the store, I take them off outside of my car and roll them in on themselves. And then I get inside my car. And so I'm only using them outside. And then when I get home, I wash my face. Because it's hard surfaces, I had one of my parents say that they'd heard, oh my gosh, you can get this from gas pumps. We have to be very, very careful around gas pumps. Gas pumps are hard surfaces, and like any surface, they're just as dangerous as other hard surfaces. So door handles, flushings on toilets, petrol pumps, anything that's a hard surface that people are touching frequently potentially has the virus on. The way you deal with it is you wash your hands to remove the virus. You can also clean these surfaces with disinfectants. Remember, there's that soap bubble around the virus that's very susceptible to getting broken or burst. So there's lots of things that work well. Alcohols. So a 70% alcohol solution, like you may see if you have a swab, you go to the doctor's office and you're getting an injection, they'll swab the skin. They're swabbing it with alcohol. That works great. Quartz or quaternary ammonium compounds are great destroying membranes. Those are found in things like formula 409. And bleach is fantastic. Bleach is about the best, but it's damaging to surfaces. So use it if it's appropriate, but don't feel you have to use it. What you want to do is you want to take a household bleach and you want to dilute it 1 to 10. It doesn't need to be any stronger than that. It just takes a few minutes of exposure. If you're interested in finding out more about what you can use and how you should use it on surfaces, the CDC has a whole website on this, and this is the link. But if you just type CDC coronavirus, you'll be able to get to all of the CDC resources and see what they recommend. And this is really written for people who are just going about their day-to-day lives. How do I keep myself safe? So this is a picture of a coronavirus, and this is why we call it a coronavirus. If you look, this is that bubble I told you, the membrane of the cell around it, and these are the spikes. These are the receptors around the outside that target our cells. And if you imagine what that looks like from a distance, it looks a little bit like a crown. So we call it coronavirus, which is the Latin word for crown. So that's where the name comes from. If we were to take a coronavirus and slice it in half, this is what we'd see. So this red around the outside, that's the membrane that comes from your cells. So it's your membrane. Then they have this thing, it's called the spike protein or the S protein. And this is basically a key that fits in the lock of your cells. So you have a receptor, a protein on the surface of your cells, which is an exact match for the shape of this spike. So imagine it's a bit like Velcro. If the Velcro is not exactly right, it's not going to stick. But if it's the right kind of Velcro, it'll stick very, very well. So you may hear a lot about the S protein in the press because the S protein is what we're going to use most likely as a vaccine. There's another protein, which is the hemagglutinin esterase, which is a complicated name, but this is involved in getting the soap bubble to leave the cell. So the reason this virus is able to infect so many people is it's able to leave your body and get to somebody else's body. And in order to leave your body, it has to take that soap bubble off the surface. So if you can imagine what we drew before, where we had a virus budding from the surface of your cell, you can imagine it's kind of hard for that bubble to pull apart from the soap bubble it's attached to. That's what this protein does. It basically chops away the cell membrane, the bubble, and allows it to escape. And then inside is the genetic material. So this is the RNA. And this is actually really good for us because the coronavirus RNA is a single molecule, which means that it doesn't go through any kind of genetic reassortment. If you think about the flu, every year you have to get a different flu vaccine. And that's because the flu is made up from lots of different pieces. So let's say you had flu A and flu B, and you had all the pieces from flu A and all the pieces from flu B, and they got into a single cell. The virus that emerges, it's like shuffling a deck of cards. You can pick up some of A and some of B, and you end up with a completely different virus, which is why we have to have a vaccine every year to take into account this assortment. That doesn't happen with the RNA, the material within the coronavirus. It's a single piece, so we don't get that genetic reassortment. And it also appears to be very, very stable. I think they've recorded up to 23 changes over a large piece of RNA since it first emerged, which is an incredibly stable thing. That's important because it means it's not going to change much. So if we come up with vaccines, they should work. It also means that it's unlikely to change in the way it makes us sick. So some people have worried that maybe this thing will mutate, become a lot more dangerous. And if it becomes a lot more dangerous, it could be a lot more problems. That's unlikely to happen because viruses evolve to make you less sick, not more sick. But this one is stable, so we don't expect such changes in that. This is just to show you the size of coronaviruses. So this is the coronavirus over here. It's actually a big virus, which is also in our favor. It's 120 nanometers, which is a scale that probably doesn't mean a lot to most people. But the way if you want to visualize how big this thing is and why you can't see it, imagine you have a grain of sugar on your finger. You've got a single grain of sugar. Now imagine that single grain of sugar on your finger the same size as a two-story house. If that grain of sugar is the same size as a two-story house, then the virus particle is the size of the sugar particle on your finger. So you can imagine you've got a sugar particle. If I can draw one like that, it's pretty small. Let's say it's about 1 millimeter across. If you make that a two-story house, then an actual grain of sugar is about how big that virus is. So it's incredibly tiny for us, but it's actually quite big as a virus. It's not as big as the biggest virus. That's the virus that causes smallpox, but it's still a pretty big virus. And that's actually, again, for our benefit. And the reason for that is it's possible to filter out and trap it. So this is just a visual of how big the virus is. So imagine that this is a strand of human hair. So if you imagine you take one of these off your head, how big that is, then this is the approximate size of a coronavirus particle. A surgical mask has a filter size about this big, and an N95 mask has a filter about this big. So it would seem that the virus can actually go in quite quickly because it's much, much smaller than that hole. So if you were to breathe air in, it would go through. But it turns out that the virus doesn't come away in individual particles like this. It actually comes away in droplets. if you look at someone who's working in healthcare and is dealing with the coronavirus, remember we talked about mucous membranes and how it can go in through your mouth, your nose, and your eyes. They've actually got a lot of protective covering. They probably have 2 masks on underneath. They have a protective suit over the top, and then they have goggles. And the primary role of goggles and face shields is if someone sneezes or coughs, they don't get that material in their eyes, and they also don't touch their eyes. But you can actually protect yourself because of droplet spread. So it looks like the virus is primarily spread in droplets. And so droplets are formed basically when you cough or you sneeze. So rather than those individual particles that would be impossible to see, you actually get these tiny, tiny drops. And some of these might contain thousands of virus particles. They're also kind of made of spit, and they're heavy, and they fall out of the air. And they're really quite large. So even a mask that you make out of cotton, this is some masks that we've been making for local hospitals. So if you have some cotton and you have 2 pieces of tight weave cotton, something like a t-shirt, and you sew them together, that will give you enough filtration to keep most of those droplets out. And it's been shown that they can reduce the infection rate by 70%. So when coronavirus started, there was a lot of information about don't wear a mask, don't wear a mask. And the reason that we put that information out is because we didn't want people using supplies that needed to go to hospitals. And the worry was that if people started hoarding masks, then the people who needed them in a hospital because they were being exposed to coronavirus patients weren't going to get them. Now, because there's such a high load of coronavirus in the population, it really is worth protecting yourself. And so wearing a mask can be really important in doing that. But it's really important that you use it properly. If that mask is working well and you've got it against your face, all the virus particles are going to get trapped on the surface. So if you put that mask on and then you take it off by grabbing on the front, then you're going to be putting the virus particles that you captured in that mask on your hand. Then you might be spreading them around. So you've really got to think about the mask as protection. And you want to think about if it's doing its job properly, the front of it is going to be infected. Now, if you have the virus and it's doing its job properly, then the inside is going to be infected. But in either way, the virus itself is going to be possibly contaminated with this virus. The mask is going to be possibly contaminated with this virus. And you have to make sure that you don't in turn use that to make somebody else sick. So don't wear them day to day to day. Wear them one day and then wash them. Have a couple of these that you have. And then just be careful how you use them. This is someone wearing a mask, and you can see there's some pretty big gaps around the side of the mask. You want to make sure that you have a snug fit on the mask. Otherwise, when you're breathing, you're actually going to be drawing air in through the side of the mask. And then as the virus walks by, you're actually going to suck it in the mask and then spread it out. So you're actually increasing your possible exposure to the virus. So you want to make sure that your mask fits well. You don't want to touch the front. You want to take it off. You want to replace them frequently. You really don't want to do this because if there's virus on the front of that mask, you basically smeared it all over your face. So masks work really well. Just imagine that they're dirty when you're done with them at the end of the day. You want to wash them. You want to get a new mask. A lot of people ask about where the virus may have come from. And we call the source a reservoir. So where did the virus emerge from? So I recommend anybody who's interested in this. There's a book by David Quammen called Spillover. That was really well written. As a microbiology, I thought it was really well done. And the definition of a spillover event is when a reservoir population containing a pathogen or an organism that can make us sick comes into contact with a new host population and transmits that pathogen. So in this case, you have an animal that is sick with something that we've never seen before. It comes into contact with the humans and it transmits it. So that's called a spillover because it's spilling over out of the animal population into the human population. And there's been a lot of spillover events. You've probably heard about the Ebola epidemics, a hantavirus that was a big one in the Four Corners region. SARS, which is subacute respiratory syndrome, severe acute respiratory syndrome. And that was cousin to the virus that we have right now that emerged in 2003. And then you have MERS, which is Middle Eastern Respiratory Syndrome, which emerged in 2013. That occurs in the Middle East. But there are other organisms that you're probably familiar with that you didn't realize came from spillover events, so came into the human population from animals. A lot of these came in during, medieval times when we started farming. Bubonic plague, the Black Death, that comes from gerbils in North Africa. Tuberculosis, that probably comes from cows. Smallpox also probably comes from cows. And human immunodeficiency syndrome, or HIV/AIDS. We thought that was a relatively recent spillover, 1979, into the human population, but the genetic evidence suggests it spilled over into the human population as far back as 1890. It just took that long to take off as a pandemic. And the reason it doesn't happen very often, so we know that all mammals have coronaviruses. It doesn't matter if you're looking at your cat, your dog, or your gerbil, they have coronaviruses. It doesn't spill over very often because a lot of times the virus has adapted really well to infecting its hosts. So in the case of SARS, which is severe acute respiratory syndrome, it originated in bats and it was really, really good at infecting bats. Humans look too different from bats. So, and I got humans as Ryan Gosling here, but humans and bats look too different. But what happened was the bat was able to infect, this is a civet, which is a mammalian species in China. And the civet looks enough like a bat that the virus could adapt from the bat into the civet. And then it replicated in the civet for a certain period of time. And unfortunately for us, the civet looks enough like a human that the virus was eventually able to jump from the civet into humans. So it's called a species barrier because it's not possible for the virus to go directly from the bat into the human because we look so different. But what we can do is we can have an intermediate host that allows the virus to jump. So for SARS, it was a civet. There's some research, this is a paper that came out, I think in 2005, that showed that the bat form of the virus was only 88% identical to the virus that makes us sick. And it was unable to replicate in the cells with the receptor we have that coronavirus attaches to. However, it was 93% identical to the civet. So that's how we think we have evidence that it jumped from a bat into a civet and then into a human. For Middle Eastern Respiratory Syndrome, we also have a bat that was the origin. Again, the bat wasn't able to infect a human, but it was able to go from the bat into a camel that was similar enough for this virus, and then from the camel into humans. So that's Middle Eastern Respiratory Syndrome. What about COVID? So what do we know about COVID? We know that it came from a bat. Genetically, it's very, very similar to bat coronaviruses to the point that it really couldn't have had an origin anywhere else than a bat. What we don't know is whether there was an intermediate species or not. There's been some publications that suggest that a pangolin might be the, might be the host. So pangolin, this is very cute animal here, that it may have jumped from a bat to a pangolin to a human. But there's been a lot of indications that the virus can infect ferrets and it can also infect cats. And so there's a famous case of it infecting a tiger in a zoo in New York. And I've heard that the only reason that that particular tiger got tested, it was the only one they could get close enough to. So we're not sure if there was a species barrier, if it jumped directly from bats into humans, or it went through an intermediate species. Right now, something that maybe looks cat-like or ferret-like is our best guess. This is just a figure I like to show about coronavirus. And everybody feels like this is just such a terrible thing that's happening, and there's way worse scenarios. As microbiologists, ever since 1918, we've been wondering and worrying about pandemics. We thought it was going to be Ebola. We thought an Ebola pandemic was possible, and about 5 years ago, it looked like that might have happened. The reason you don't want to have Ebola is that it has about a 43% mortality rate. So 43% of people, 44% of people infected would die. So this is a sphere that shows the total number of infections and the total number of deaths from Ebola. And that kind of death rate is getting up to close to the level of Black Death. And that would be a society collapsing infection. But Ebola doesn't transfer from human to human easily. You've actually got to have physical contact. It's not airborne. And so even though there is a high mortality rate, as soon as you were able to get people away from each other and you have social distancing, you could basically knock out this virus. There are two other coronaviruses that we've been worried about. One is the SARS, the severe acute respiratory syndrome, which emerged in China in 2003, and the other one is MERS. SARS had a much higher mortality rate, about 10%, but it didn't really spread from human to human. There were very few transmissions. It was very difficult for it to spread. The other one was MERS, and that was really quite a scary infection. That was 35% as a respiratory virus, but you had to get it from a camel. It wouldn't spread between people. You actually had to get it from a camel themselves. And so that was a high mortality rate, but a really low infection. Now MERS, and this is, I think this number here is off because this was a figure from about 2 weeks ago. I think we're over 2 million now. The death rate is about 4.5% in this figure, but that turns out not to be really the case. The problem we have with COVID is the amount of testing. So the way you understand this virus is you have to understand how many people have been infected. And in countries that don't do a lot of testing, like Italy and Iran, Spain, and particularly the US, we aren't testing enough people. We're only testing the very, very sick. And because we're only testing the very, very sick, we tend to be selecting for a population that has a greater likelihood of dying from the infection. So that's what pushes the mortality rate up. If you look at countries that are testing a lot more of the people that of their population, Germany has a mortality rate of 0.4%, which is getting it in the range of a severe flu. And that's because they test everybody. And it turns out that as many as 50% of the people that actually had the infection don't even know they've had it. The symptoms are so minor that they never even thought they were infected with the virus. So until large-scale testing comes online, we won't really understand what the mortality rate is. The reality is we probably won't understand it for a few years. The other question I get a lot of is why did this emerge in China? And I've actually spent quite a bit in China, a bit of time in China. This is me at one of the villages that we stayed at. And the Chinese people are some of the most gracious, honorable people you've ever met. You have to be very, very careful when you go to China that, you know, if they ask you if you're hungry, you have to say, no, I'm full. Because if you say you're hungry, they will give you food, even if they don't have enough to eat themselves. It's also a very, very poor country. We spent a lot of time in the villages, and an entire community can be based on one family that's lived together and built this village together. It's essentially one house. They're all farmers. And the wealth of these people is basically bound up in the animals that they have. So they're overprotective of their animals because they don't really have banks. What they have are livestock. So this is the building we were sleeping in. This is our bedroom over here. And this is where they kept the muskox that they used to plow the fields. And the chickens were there as well. And you'd get up in the morning and the chickens would jump up from below and then they'd run into here, and you'd wake up in the morning and there'd be a chicken running around in your bed. And there'd be rats in the ceiling. Anyway, there's a lot of interaction with animals in ways that we don't have interaction with animals in the West. They have very poor levels of hygiene. They don't have indoor plumbing. This was our cook, and she's made some tofu from soybeans from scratch for us. You can see her boiling the tofu here. And this is her water. She doesn't have running water. They get their water from a cave entrance. They have a cistern, and she'll go out and she'll get a bucket of water. If I wanted to wash my hands or wash my face, I used to have to take a bucket and go over to the cistern and get a bucket, find a bowl, and do it. They don't have any running water. So the level of hygiene is lower simply because they don't have access to the quality of plumbing that we have in the West. The other is that they have these open air markets, and they have a lot of exotic food in these markets. And once you've been to China a few times, you kind of get used to scenes like this. But if you've never experienced it before, it can be quite shocking. So this is a livestock market. So it's just some things to point out that, you know, when you're in China, you expect this, but it can be a shock to some people. So here you have a chicken sitting on some greens. And so the chicken is probably going to be sorted and sold, and the greens will be sold even though the chicken's been sitting on it. You have some live frogs that were caught wild. The chopping board that's not getting washed over here between different species being slaughtered on it. You have a bucket here with lots of different species being slaughtered. They're just wearing cotton gloves to do this. There's no way of washing their hands in between. So you have a bucket full of the blood and organ of lots of different species. They're weighing things in a bucket that doesn't get washed and on scales that don't get washed. And there's also a culture that a lot of these animals have powers that you can get by eating the meat raw. So livers and hearts and internal organs eaten raw give you strength and vitality. And so they'll actually eat a lot of raw meat. So because of this, they just get exposed to a lot of viruses that we wouldn't. And they also, because of poverty in China, any kind of protein is important. I remember when we were staying with the farmers, and they'd have a big bowl of stew for us, would there be a fight with the chopsticks to try and see who could, just between us, the cavers, the locals were much more refined, but somebody would find a chunk of meat, and then we'd all go fighting around with our chopsticks because it was so rare to get meat with your dinner because it was so expensive, and they very rarely get protein in their diet. So anything that they catch out in the wild, they'll take in the market either to sell or they'll actually consume themselves at home, just simply because it's protein and it's too precious to lose. And because of that, they also get exposed to lots of different animals and the potential for lots of different viruses. So this is just a list of the animals that were available to buy in the market in Wuhan. So you've got really normal things like cattle, pork, and poultry. Got koalas. Dog is really common in China. Snake, turtles, civets, crocodiles, giant Chinese salamander, bugs. It's cicadas. Yeah, anything that's protein gets eaten. So I want to briefly go into the epidemiology. So we've seen something very similar to coronavirus, and that was in 1918, and that was the Spanish flu. We're not quite sure where the Spanish flu came from. We think it started circulating somewhere in France during World War I in maybe 1916 or 1917. But this is a picture from America, and you can see everybody is wearing their mask as well. So this is starting to be a scene that looks familiar, but this is kind of cool. So they actually had a mask on their cat. And then I don't know if you've seen, but my mailman did have gloves on, but he's starting to look like this. So we've been through these kinds of epidemics before, and there's pretty standard measures for limiting the spread. The big one is social distancing. So this is an outbreak of the Spanish flu in the United States in 1918, and it was the response of two cities, Philadelphia and St. Louis. One city carried out social distancing and the other one did not. In Philadelphia, they didn't carry out social distancing, and they had a very dramatic peak in the number of deaths very quickly, and they overwhelmed the healthcare system because of that. In St. Louis, they actually carried out social distancing, and you can see that the total number of cases was lower, and the total number was also spread out over a much longer period of time. And the reason it's important to do that is because every day we understand this virus a bit more than we did the day before. And because of that, medical treatments are getting better. So your chances of surviving increase if we can push this arrow down the road. So the further we can go, the more time we give ourselves to come up with antiviral drugs, treatments that increase your likelihood of survival, and even vaccines. So the reason we call it flattening the curve is we want to decrease the number of total infections and how quickly they form. And this is related to R naught. And R naught is basically how infectious an organism is. So the idea is if you have an R naught of 2, so an R naught of 2, it means every infected person will infect 2 people. Have something like measles, and every infected person infects 18. And measles is so bad that if somebody walks through a grocery store with the measles, there's a good chance that everybody susceptible to the measles virus in that grocery store will be infected. So that's why it has such a high R naught. We think that coronavirus COVID has an infectivity about 2.5. So we can model how that works for reducing infections if we're going to use an R naught of 3 here. So the idea is that every person who's infected will infect 3 other people. So let's say this person is infected, and then because of social distancing, on this day, they decided to work from home. That means that they didn't infect this person, these people didn't get infected, and a result, and you can see that at the end of it, all those people didn't get infected because this person decided to stay at home one day and not infect this person. And then the next person decided not to go to a barbecue, and then this person didn't get infected. You have more people that weren't infected. This person didn't take a flight. This person stayed home for an extra day. And you can see how we can quickly reduce the total number of infections just by reducing how many people you're interacting with, reducing how quickly that spreads. So if you can imagine that if you have 2 to the power R naught, so basically if it's 2 to the 2, you'd have 2 people, then 4 people, then 8 people, then 16, or 2 to the 3, you can see how quickly it spreads. And what social distancing does is it reduces this number. If we can get that number below 1, what happens is the virus starts to go extinct. And that's what social distancing does for us. This is just a graph showing the impact of social distancing. This was based on a model between the total number of cases if social distancing changed by 1 day. So instead of starting on day 20, started on day 21, there were 40% more cases just because people interact less. So that's why we're doing it. And this is the really good news. The last time I presented this, we were out here somewhere, so it didn't look great. But you can see we started to flatten the curve and we started to drop it. And that means that social distancing is really starting to work. And everybody's like, this is great. So what's the next step? When do we get back to normal, and how do we get back to normal? So there's a lot of things that have to work for us to get back to normal. And the big thing is going to be whether the virus is seasonal and whether we can get testing online. So what does seasonality mean? Coronaviruses, I said, are RNA viruses, and RNA is really susceptible to temperature, and it's really susceptible to sunlight. So there's actually a coronavirus season. So from December to April is the big coronavirus season. And 30% of the common colds that we have are caused by circulating coronavirus. There's a good chance you've had a coronavirus in the last couple of years and you just got a cold. This one's just a bit more severe. Because of this seasonal period, we're hoping that there's a window for us from May to October where the virus basically goes quiet and we don't see it. It's still going to be spread in the population, but that R naught value is going to be so low that it's going to kind of simmer along underneath, and we're not going to see the explosion in infections that we did start to see in March. So that's one thing. We don't know if the virus is seasonal yet. We don't have enough data. It's all going to depend on what we see happening in warmer countries. Certainly places like Australia and Brazil have cases. They just don't have the kinds of cases and the kinds of numbers that we see. So if you look at the Northern Hemisphere, you can see really dramatic cases, but not so much in the Southern Hemisphere. So maybe there's an indication that it is seasonal and it will kind of go away. It does mean that those Southern countries are going to be susceptible and in danger over our summer months. The other thing that we need is herd immunity. So right now, nobody has immunity. So if somebody is sick, they're They're going to pass it to some people who are completely susceptible to it. They have not seen the virus. We're all completely naive to this virus. It's brand new. It's never appeared before. So they get sick and then they make other people sick. And you can see there's nothing to prevent the spread. Partial immunity can be brought about two ways. One is that social distancing means that you prevent those interactions, and that's how you reduce the cases. The other is that this person has been exposed and recovered. So now this person doesn't pass it on. So what we're going to see is we're going to see partial immunity where a certain percentage of our population is already going to be immune to it because they've already been exposed to it. And there are some estimates I've seen anyway where from 3 to 30% of natural immunity by the end of this outbreak. And if it's close to 30%, we could be seeing life return to normal in May. The final thing is herd immunity, and this is where we want to get to, where so many people are resistant that even if one person infects another person, there's nowhere for it to go. So either these people have been immunized or they've already had the infection, that the level is so high that no more people can be infected. And that basically shuts infections down. That's how we mostly deal with influenza or try to control influenza. It's certainly how we control measles and why we don't have large measles outbreaks. I think our herd immunity rate, so how many people have been vaccinated and are resistant to measles, is about 97%. So the idea is if you have a huge population of people and you have some people here who are infected, all of these people here are resistant to infection because of that herd immunity. So even if you have a couple of susceptible people here out in the population, they're never going to get infected. So that's what we really need to control this thing. The other is tracing contacts. So this is the idea that, we have one person who infects another person, and then what we need to do is we need to look at all of their contacts and all of the people that they came in contact with. And we have to trace everybody. So the idea is if we find somebody who was infected, we can identify people who have the infection and we can say, can you please stay at home for 2 weeks and not go anywhere? And then these people, because we've contained them, instead of the entire population being asked to stay at home, it's just a few people that we're asking to do that. And the level of disruption becomes a lot less. In order to do that, we have to know who's infected. And for that, we have to have testing. The initial testing was done, you may have heard of this term, by reverse transcription quantitative PCR. It's quite technical. It's quite difficult to do. It needed some specialized labs and it wasn't automated. We were able to start testing as early as January 16th, but I don't believe the US began testing until March. And certainly we were far behind. I don't think we got anywhere close to, I think South Korea has been testing 2 million people a week. And at one point in the US, we were only testing 10,000 people a week. So we have to get to those levels, because when we get to those levels, we can test people and say, you have the virus, please stay at home. And that's how we control the infection. You might see things, yeah, and the other thing is the way we test, this is how they do a nasopharyngeal swab. This is how we used to have to test for the virus, is that we thought the virus replicated way back here. And because of that, people said if you got tested for it, it felt like they were scratching your brain with the swab. Lots of people have said that they've noticed their smell disappears when they have the infection. And the researchers followed that and figured out that you actually have replication way down here in the nose. So now instead of swabbing way back here, you can actually do a very convenient swab on the tip of the nose. Expect to go places and have people take your temperature. Temperature is the first sign and is often before you start shedding virus. So if people are taking your temperature 10 times a day, thank them for doing that, because it means that you'll know as soon as you can that you need to go home and rest and recover, and also seek doctor's treatment. As I said, there's more of a rapid screen because we don't have to sample so deeply. And we also have the ability to test people's blood now for antibodies against the virus, which makes us understand much more quickly if someone has been infected. And that would allow a serology test can be very quick and could be as little as 10 minutes. And the idea is a serology test could get to the point where is somebody who's going through the airport at TSA could get tested before they go through security. And by the time they've cleared security, we would know if they were positive or not. And then we could stop that person getting on a plane. So there's lots of things that we can do to limit the spread and get everybody back to work and the economy going again. But it's all going to come down to testing and our ability to identify people who are sick. So I've actually been quite optimistic. You know, microbiologists have always kind of thought that there is another pandemic brewing. And when we saw Ebola, everybody got nervous. And when we saw SARS, we got nervous, and MERS, everybody got very nervous. But there's a lot of things to be optimistic. We were able to identify this virus very early. I think the first reported case in China was December 31, and January 16th, we were able to test for it. As I said, the virus doesn't change very well, so there's a low mutation rate. It means that we should be able to come up with effective vaccines. We don't know if there'll be some change to the virus over years, in which case we might have to get a seasonal vaccine against it, the same way we go against the flu. But, you know, at least we have that low mutation rate. I said relatively low mortality simply because there were much worse coronaviruses out there that we're worried about. And if that mortality rate is 1% or lower, then, you know, we really have dodged a bullet here. We also have had the SARS and MERS models. So when SARS emerged in 2003, the microbial world took a collective breath, and a vast amount of money was invested in understanding the virus, how to create vaccines, and how to create antiviral drugs. So we have all that body of knowledge to work with. This is not a virus that we've never seen before. It's very similar to SARS, so we have a lot of information already to get going. And the other is seasonality, which we're not sure about. But if it is a seasonal virus, then it should start to die back and just kind of fizzle along in the background until the fall. And so the other thing I want to say is that we've done this before. The 1918 Spanish flu epidemic looks an awful lot like what we're going through right now. And we didn't have the kinds of the tools and technology that we have available to us now back then, and yet we were able to come together and we were able to beat this through good epidemiology, good ways of dealing with the epidemic, just what we've done with the social distancing and trying to protect our friends and neighbors, be careful about our parents, protecting the vulnerable. So we've done this before and we've made it through. Just gonna, if anybody is interested, I'm gonna recommend some books. These were written for the general public. The one I've already mentioned is Spillover. That was really interesting. It's all about how viruses can spill from, and bacteria can spill from animals into humans. It's not good reading for the faint of heart. There are some pretty bad viruses out there. Guns, Germs, and Steel by Jared Diamond was a fantastic book. It's talked about how not just technology, but also infections have changed the course of human civilization. And then another book's very similar to that is 12 Diseases That Changed the World by Erwin Sherman. That's a fantastic book. It really describes how, you know, like things like the Irish potato famine, you know, basically changed the culture of that country and also the United States. There's a couple of podcasts that I listen to. These are very technical. These are for experts in the field, but if you're interested, they try to make sure they explain things for people who don't have a background in microbiology or virology. And then just to tout the lectures, the courses that I teach at Akron, I teach an Introduction to Microbiology course, which is kind of a general microbiology course. And I teach the Advanced Medical Microbiology course, which is more prepping students to go to medical school. And also in the medical microbiology program, we have one of the biggest rates of students going into the medtech program. So a lot of the people in the state of Ohio right now who are doing some of the tests for the coronavirus are actually students that came out of our microbiology program. We are thinking about making the introduction to microbiology kind of an open course, maybe something that's online, mixed online, and maybe a week of lab work for people that are interested. So keep an eye on that if you're interested. And with that, I know I said I was going to keep it short. The last time I did this, it was an hour and a half, so that was an hour. But okay, Randy.
So hi everybody. I'm just been lurking here.
I've been recording a whole bunch of questions here. The first and most important one is, are you recording this? Many people will think that it's great and that they'd like to be able to listen or look at it again.
Yeah, so we recorded it, and Akron is going to put it up on the website. I did a much more, like I said, a longer lecture that's up on YouTube, and we can put a link up for that. But this one will be recorded, and the university is going to make this available.
Super. There's a mix of questions. I'll just sort of go through them. There's a question about there seems to be a resurgence in some of the Asian countries, and a question partly, I think, about why that's going on in the country, but also whether a person who had COVID might have a recurrence.
So the second wave is something that we're going to have to expect here. There's no way we can avoid the second wave. How bad it is and what it looks like is going to depend almost entirely on how many people have immunity to the infection. So expect that. Don't be concerned. A second wave is part of it. That's where we have to get testing, because you can identify those cases and get those people isolated. So what we're seeing in these countries is that second wave, and to be prepared for it and emotionally ready, and not to think that we're never going to get through this. This is just part of the cycle. There are 5 phases that we have to go through it. In the second phase, the first was the initial infection. The second is going to be the socialization. The third is going to be the second wave. So it's going to happen. It doesn't mean that these people don't have immunity. There's nothing to suggest right now, based on a lot of the antibody studies, that people don't become immune to this. That's not saying the virus won't change over time. So there can be some genetic drift. So there can be some subtle changes to the virus so it escapes our immune system. That doesn't mean you're going to get as sick as if you were exposed to COVID the first time round. And it also means that there'll probably be a seasonal vaccine that you have to take if we see that happening. But that's something we'll have time for. You can see that coming. We can plan for that the same way we can plan for the seasonal flu. So I don't think anybody's expecting anything less than these second waves. The thing is, you have to write it out and you have to expect it. And you have to know that that's just how just how these things work. But every time you get these waves, you can isolate, you can test, and you can get people quarantined.
Super. There's a question about what's the role of fever? Is it a way of fighting an infection? If that's the case, why would we want to lower it down? And what's the problem with having too high of a fever anyhow?
So this is like the question that microbiologists and immunologists get. And the answer is we don't know. You know, you'd say with 70 years of understanding this, we don't really know. We know inflammation is very bad in the infection. A lot of the problems that patients have, the difference between getting sick and recovering and getting sick and getting worse is actually your own immune response. It's your immune response that does this to you. And it's the inflammatory response that seems to be doing that. So you really have to be careful that, you know, you eat well, you know, you don't overdo it on the sugar and the alcohol, you know, you get plenty of vegetables so your immune system's in good shape. But we think that inflammation causes, as you suggest, an increase in temperature, and that increase in temperature makes it harder for the virus to replicate. And there is some evidence of that if you do this work in cultures, but there's also effects on our cells. And so it's very hard to tease these things apart in the laboratory, and we can't really do it in humans. The reason you want to lower your fever is, one, it's that inflammatory response is quite bad in this virus. So it's designed to clear the virus, but it seems to be having a really negative effect in overall outcomes. You want to try and avoid NSAIDs, so things like ibuprofen, and manage it with Tylenol. But at the same time, you shouldn't avoid ibuprofen. You know, if you feel terrible, you should make yourself feel better. And that's part of what we're doing, is if you have a big fever, you know, you're going to feel awful, and bringing that down helps. Was there anything? No, I think I'll just leave it at that. Talk to your doctor. But yeah, you do want to bring a very high temperature. If you get above 106, it can cause a lot of problems. I think it's 105.5. You can get alopecia, which kills the root cells in your hair, and you'll actually lose your hair. So you do want to be careful about not letting that fever go too high.
I think this is a sort of a good question for the very beginning. What's the difference between prions and viruses?
That's a great question. Prions are just proteins. There's no genetic material. And prions are actually formed by a misfolding of our own proteins. So it can happen spontaneously through genetic mutation. And there'll be a familiar, it's called Creutzfeldt-Jakob disease. So it's actually, it goes inherited generation to generation. You can get a prion if you eat a contaminated meat. There's a big species barrier. It seems to be a spontaneous mutation that's quite common in sheep, causes a disease called scrapie. But if we eat sheep, because of that species barrier, we can't get sick. However, if a sheep is fed to a cow, it could overcome that species barrier. And then when we eat a cow, we can get a prion. So that's where the mad cow disease outbreak came from. We've managed to get that out of the food chain now by making it illegal to feed dead sheep to cows, and so that's gone out of our food population. But it's actually a really, really interesting field of microbiology. And as I remember being an undergraduate when Stanley Prusner first started publishing on it, just having my mind blown that a protein that's misshapen can bind to a healthy protein and cause it to become misshapen. And then you have a cascading effect that damages all the proteins and in the process damages your cells.
Yeah, that's crazy stuff. A couple of people are interested in viral load, partly how much virus has to hit you for you to get sick, and also where it is. Is it in the nose or in the throat where it's most abundant, and how that might relate to the SARS-MERS comparison?
Yeah, we don't know. We suspect that there is a dose dependency. The reason we think there's a dose dependency is because what we're seeing with healthcare workers, so they're probably going to be exposed to much higher levels in the healthcare setting. And in turn, we think that's related to their immunological response. Like if you get too much of this thing and it keeps hitting the immune response again and again and again and recreating that inflammation, and that drives that kind of negative feedback loop that causes all the damage in your lungs. So we think that there's definitely a dose dependent response. We don't know what the minimum infectious dose. We wish we did know, because if it's a few thousand, it's a lot more difficult to pick up than if it's one. Because if it's one, it's kind of like measles, somebody walking through the grocery store making everybody sick. So there's probably a minimum threshold that you have to be dosed with. If it was very low, I think we'd be seeing an awful lot more people infected. But again, we haven't got testing online, so we don't know what that number is. I'm going to say the numbers I've heard, I think there's over 500,000 cases in the US right now. And I think we've got a population of 370 million, so it's less than 1%. And the estimates that I've read for actual infections are somewhere between 3 and 30%. If it's at 30%, then yeah, that's going to be a very low threshold. So, but we wish we knew that, and it's the first question the virologists are asking. It's hard to do. So a lot of the tests are based on that piece of genetic material, the RNA test. And you see a lot of people publish on RNA, we found RNA, we found RNA. That's very different than an active particle that can make you sick. I got interviewed for a newspaper today where they were like, well, they found RNA here. And it's like, it doesn't mean anything. It doesn't mean that there was a particle that can make you sick. It just means some residual part of that virus was there. So there's a big difference between enough to make you sick and the genetic material. But that is a great question, and that's the one we're still asking.
So there's a sort of follow-up from what you just said. Someone mentioned the claim about COVID being detected on shoes maybe 5 days later. Is that exactly?
Yeah, it's just RNA. RNA, these viruses are really weeny. I mean, with that membrane, they're kind of weeny. So just because you detected the RNA, it's unlikely. We know it's going to be on door handles. We know it's going to be on petrol pumps. We know, anything that people's surfaces people are touching, we know it's going to be, and you have to wash your hands and don't touch your eyes. As far as on your feet, I mean, that study was done in hospitals, and people work on exactly this same kind of surface. They weren't working across grass. They weren't walking on concrete. They weren't getting in and out of their cars. And I don't know about you, but I take my shoes off when I come in the house, and I'm not in the habit of touching the bottom of my shoes and then touching my face. So I had a very panicked email from someone, and they bought this sliced meat at the grocery store. And they'd heard that the virus can only survive 3 days. And they wondered if they should leave the meat out for 3 days to be not sick from the virus. And I was like, okay, first of all, we have hygiene in the grocery store so you don't get sick from food poisoning. That same hygiene will protect you from the virus. And please put your meat in the fridge. There's a reason we do that, because you'll get sick from food poisoning. So you have to be pretty reasonable. You know, it's about wash your hands and don't touch your eyes. And now wear a mask. You should be safe.
That's good. A couple of questions here sort of about defenses. So there's one that's talking about, it seems that we can defend against bacteria a little easier than we seem to be able to do by developing vaccines. There's also some about maybe using other viruses to compete or fight against them, and some questions sort of involving herd immunity, and can one person become herd immune, or is this something a little different than that?
So as far as bacteria, it's much harder to make a vaccine against a bacteria than a virus, because viruses have one protein that they use to attach. So a lot of the great vaccines we have are actually against viruses. We do have some vaccines against things like tetanus, which is Bacillus tetani. That's not a virus against the bacterium. You still get sick with the bacterium. The vaccine is actually against the toxin it makes that makes you sick. So you actually block that protein. So it's much, much harder to make vaccines against bacteria. There are some component vaccines, but it's actually quite difficult. And I don't want to scare anyone with what's out there in the bacterial world. I mean, it's scary enough with what's in the viral world, but there's things out there as scary in bacteria as viruses. And, you know, we are, just like we've been working on SARS and Ebola and all these other viruses, you know, one of the things we work on are these bacteria. Trying to remember all the parts of that question. So as far as herd immunity refers to the population as a whole. So it's all about breaking down those contacts. So if this person's sick, those 3 people around them that they could possibly make sick, if they're all immune, then the virus dies. And so you have to get those numbers up so that maybe if 2 of those people are immune but 1 isn't, then it's going to spread. So we've got to get those numbers up. The way we've been doing it now is we've just been limiting contacts. And so that is social distancing. The next thing to do is identify not just a sick person through testing, but every single person that they've come into contact with in the last 2 weeks. And that can be like 200 people. But if we can do that kind of screening, and the government's talked about getting like 150,000 people online to be able to do that kind of tracking down and then test. What you can do is you can find those 3 people that got infected and you can ask them to just stay at home. And that's how we kind of keep this thing under control until vaccines come along. As far as other viruses, this is really exciting news. And I think we only heard this in the last couple of days. There's a new strain of the virus. And I said it doesn't really mutate, and it doesn't. But it appears there's a strain of the virus that they found in Asia that has lost a protein. So this only has 8 proteins and it's lost 1. And people who've had that form don't really have severe illness. They have more like a cold. So one of the things people are wondering is if we can get that strain, can we use that strain as a live vaccine? And we literally will just start spraying it in people's noses as fast as we can grow it. And they're going to have a cold and they're going to feel terrible, but they're not going to be susceptible. So yeah, it is possible that we can use another strain. Much more likely are some of the vaccines that are being developed. So I think there are 3 vaccines in clinical trials now, which is like the fastest in history for ever being able to go from a new disease to a vaccine. And that's because of the work that was done for SARS and MERS. So one of them, I think, is a component. So they're actually just going to inject a piece of RNA that's our kind of RNA, not viral RNA, into our muscles and get our muscles to actually start making that spike protein. And then our body will make antibodies against the spike protein. And what they do is they'll stick to it before it can stick to our cells. So imagine that Velcro, like a Velcro covered ball, and you throw it at a surface and it sticks. Those antibodies will stick to that surface and prevent that Velcro from working. And so there's some new antiviral drugs that are coming online. There's new treatments. I think the latest I heard is that ventilators might actually cause more harm than good. That the symptoms are starting to look more like a high altitude pulmonary edema than they are acute respiratory distress. So it's like I said, if we can just push that curve out every day, we know a bit more, and every day we can save people's lives.
Super. This is a question we ought to make sure and deal with. Are certain races more prone to the virus? There's some thought out there that people have brought up of this, especially regarding Africa.
I don't know that there's been any race-specific data. I know that there are different standards of care that people are receiving, and that seems to have a big impact on outcomes, but I haven't heard of any genetic susceptibility of populations so far.
Super. Now I have to arrange my thoughts here. There's a couple of them asking about the prognosis here. You provide optimistic reasons, but the number of cases in the US that we have now, would you say whether it's being managed well? There's also some questions about international cooperation and vaccine development.
Yep. Everybody's got yeah, and we don't have a choice. We have to. And it's societally, it might seem like, what are our chances, but we have to, right? With the testing. Yeah, I don't remember. They reckon that the US economy has lost $3 trillion. It's like, okay, it's going to be really, really expensive and it's going to be difficult to do. But how much more money do we want to not spend to keep losing that kind of money? So yeah, these things are all going to be difficult. It's going to take a lot of political will. It's going to take a lot of mobilization. And it's also going to take a lot of skill, which luckily we have, not just in the United States, but in other countries. So I'm trying to think where there's 2 clinical trials in the US right now, and there's 1 clinical trial in China. And I'm not aware, there are 47 different programs to come up with a vaccine. Those are just vaccines, those aren't antivirals, and those are spread across the world. So I think that, we could have an ethical debate, but that's not my specialty, but I don't think we have a choice. We have to cooperate, especially for those who are most vulnerable.
Great. There's some questions about the changes in our understanding of the disease. There's speculation that the virus might stay in your body for 4 weeks instead of 2 weeks. Is that a likelihood?
You might be able to detect the RNA. It depends. In China, I believe that they were requiring you to have 3 tests that show you were negative for the virus before you were cleared as non-infectious. Other countries, I think, are saying 3 days after symptoms, which seems awfully short. It seems like 7 days after symptoms, if you're positive, would be a good window. But again, we have to have testing. We don't have enough about that. It's a really unusual virus in the way that it causes disease. And again, the thing that people don't realize is that it's not the virus that's doing the damage. The virus is killing individual cells at a time. What it's doing is creating this hyper stimulation of the immune system. Somebody asked about that inflammatory response. That's what kills you. That's what killed people in the 1918 epidemic. The fitter you were and the healthier you were, the stronger your immune system was, the more likely you were to die from the infection, because you mounted such a big immune response that released all these toxic chemicals to kill the virus. You actually ended up killing yourself. And so this virus, you see this window where people are showing symptoms in as short as 2 days after infection, up to 14 days with no symptoms, and then demonstrating signs and symptoms. And then those people, if they didn't start to get well within a week, then they were almost inevitably going to end up in intensive care. It's just very unusual in the way that people were reacting to it. SARS, we were able to control SARS very quickly early on. I think there were only 8,000 infections with SARS because you showed symptoms before you started shedding the virus. Whereas with this virus, you can be shedding it for a week before you even show the first signs or symptoms. And that's why it spread so quickly and so many people have become infected.
Does that include the high temperature? So you can be shedding even without a temperature, is that correct?
Yeah, absolutely. And that's why we think it spread. So that's why, the numbers are estimated up to 30%, is that so many people have been infected and they don't even know they're sick. So they've still been going into work. It's strange. It's not how viruses normally behave. Like for the cold, 3 days coming, 3 days here, 3 days going. You don't normally start shedding until after you start showing cold symptoms, and you stay at home, and you might make your husband sick, but you don't make everybody you work with sick. Well, you try not to, but Something like this, you can be completely unaware of the impact you're having.
There's a question here, I think, looking to confirm understanding. Since there are canals in the eyes that lead to the nasal cavity, can the virus travel from your eyeball right back there?
That's absolutely it. That's touching your eyes. So the mucous membrane will actually, we're not sure if it actually replicates within those cells, but yeah, it actually goes down the tear duct into the nasopharyngeal space. Absolutely. Yeah. And you have this, when you blink, I mean, our eyes there's this whole part of our innate immunity that's part of our eyes. So we have lysozyme in our tears that bursts the bubble of the cell. And we're constantly blinking, and that's washing. All of that stuff goes down the teardrops and then goes down the back of your throat. So yeah, that's where it's going.
And then there is, I'll just quote, I would like to ask a slightly silly question, if that's okay. I'm curious what Dr. Barton's opinion is on the hoarding of toilet paper in the United States.
Oh, yeah. I said to my husband when the infection started to spread in California and I realized it was person-to-person spread, I said, Phil, we need to get some toilet paper because we might be quarantined for a couple of months. And I don't know if it's going to be quarantine where we can go shopping or quarantine where we're not allowed to go shopping. So we went and got 12 toilet papers, but I don't know about anybody else, but it's still up there. We've only used about 4 of them. So I don't know what people are doing where they need to buy 1,000 toilet papers. There are some GI symptoms, I will give you that. So the virus doesn't just infect your nose and your throat and your lungs. You'll actually swallow it. So you make a lot of the virus and it gets in your coughing and you're swallowing it. And it actually gets into your intestines and you actually have the receptors for that spike protein in your intestines. So a big symptoms associated with the disease is actually diarrhea, but not that you would need, 50 or 100 rolls of toilet paper. So it was just panicking. And I try to explain to people, and I did make a comment on Facebook, the numbers, looking at the mortality rate out of China, the numbers are low. The 1918 flu had a mortality rate of 2.5%. And that's why it's such a good model for what we're looking at right now. So, it's not end of the world kind of like hoard toilet paper and hoard guns. And so that's really, really surprising. And I think it's just fear and it's just being afraid. And that's part of why I like to do this is, as a microbiologist, yes, I'm concerned. Yes, I don't want to get sick. Yes, I have asthma and I'm at high risk, but I'm not afraid. And I wish other people realize we're doing the right things. Everybody's, I went out for a walk the other day and, there was somebody coming towards me. I was about to cross the street and they crossed the street, right? So everybody's thinking about this 6 feet and everybody's doing the right thing and we're being supportive of each other as a community. And that's how you get through this. And so I'm sorry that people panicked and were afraid. I wish we could have done a better job of getting that information out. But to be honest, we as microbiologists, this has not happened in 100 years. And even we were kind of, we knew these things could happen. We could describe the epidemiology. We could map out what it would look like. And we've been doing this. There's been preparedness. Barton's been doing the, Bio Threat Reduction Agency for the military has been preparing for this for like 30 years. We just got the wrong virus. So, we were kind of caught unawares because we just didn't think we were going to go there because no one's seen anything like it in such a long period of time. We knew it was possible, but we don't know quite how it's going to look when it happens.
There's a question here about the whether the immune response that might vary with age might be a cause of the different rates of death for different aged people.
Yeah, absolutely. So there's a big trigger. So I'd say it's all about this inflammatory response. In fact, one of the big signals for whether you're going to get better or not is how your body responds with inflammation. So we have 2 arms of our immune response. One is we call it the innate, which is that it doesn't matter what you get infected with, we respond to it. So we can actually recognize viruses because something like an RNA virus doesn't look anything like what we see in our cells. So when we get infected with it, we mount an immune response immediately, and that's inflammation. The other side is our adaptive, and that would be the antibodies that we make. So if you get a vaccination, your body responds to those proteins by making antibodies, and then they in turn would neutralize the virus. And it seems to be that there's a lot of factors that determine whether you flip from this innate, this non-specific inflammatory response to this antibody response. Antibody response is what you need to clear the virus and get well. If you tip too far towards the innate response, inflammatory response, you can get really, really sick. And that seems to be what's killing people. And that switch can be related to alcohol. It can be related to age. It can be related to immune suppression. There's lots of different factors that actually tip that balance. And age is it's always been strongly correlated with a poor antibody response. So certainly, that's part of it. And then there are secondary conditions. So like I have asthma, and that means that my respiratory tract actually gets inflamed all the time. For practically very little stimuli inflames me. So I'm at much higher danger because I'm much more likely to tip towards that balance. And then you have people who have diseases like chronic obstructive pulmonary disease, and they're already kind of like struggling as it is, and then you lay this level of inflammation on top. So there's lots of factors, and they behave in slightly different ways, but it seems to be about tipping the balance. You want to tip it in your favor. So tell people exercise, eat right, avoid alcohol, and that'll keep the immune system tipped in the right direction.
One of our past students has a question that I'll paraphrase, which is that there seems to be some evidence in China that we're seeing more GI symptoms, and that maybe it's reproducing in the GI tract, with opening up the possibility of fecal oral infection. Do you know anything about that?
Yeah, so they've done some studies, and they've showed that they can actually track infections in the sewer systems in New York. They can actually measure the amount of viral RNA in the sewer systems, and they could actually track it going up with the viral load in the city going up. So there was actually a direct correlation. But they were never able to show that that was infectious particles. So it replicates within the intestine. We don't know if it can survive the intestine to come out. We have a lot of, enzymes to carry out digestion and break down cell membranes in our digestive tract. So the question is whether the viral particles can escape in an infectious form or not. And I haven't heard of any evidence that a fecal spread has been demonstrated. So right now it seems to be primarily droplets, which is great. It's not aerosols. So aerosols would be those very small particles that are kind of get into the air and suspended for hours at a time. It seems to be mostly droplets. So that's where these masks work. There was a study where somebody got on an airplane who was infected, and they traced all the contacts. And an airplane has a circulation system that if you had an aerosol would suck it up into the system and blow it all around the plane. But they didn't see that. And that's because they think it's just these droplets, and the droplets would have gotten trapped by the HEPA filter in the plane. It just meant that the people very close to them sitting on the plane were infected. So it's a good indication that the primary route of spread is going to be this droplets. Good question.
It's one of our students, of course.
Of course.
So could you talk about how the number of deaths we're experiencing from COVID is different from the flu or the 1918?
Yeah, we don't really know how many people died from the flu in 1918 because we didn't have testing. We didn't have serology. We had no way of testing. The best you could.
I misdirected you. The sort of we know this is more. It's more like the 1918. But how's it compared to our current flus? I think was the question.
Oh, current flu. I think this year was a bad year. I think we had 60,000 people die, 500,000 hospitalizations, and 40 million. I heard somewhere between 29 and 40 million infections. So it was a bad flu year. A good flu year is somewhere around 30 to 40,000. They're working. That's one of the things that Tony Fauci is always excited about, was a universal virus vaccine for influenza, so we didn't have to get vaccinated every year, and that would basically kill influenza in the United States. It would take that R0 value down to below 1, and it would go extinct. And that's been his favorite topic for years. I haven't got there, and our herd immunity a lot depends on people getting vaccinated against the flu virus. But it was a bad year this year, mostly because we didn't have great coverage. So they have to guess at what the virus is going to look like genetically. And sometimes they make a great guess, and sometimes they miss. So we had a bit of a miss this year. I didn't think our coverage was that great. It was about 57%. So even if you had the vaccine, there's only about a 57% chance that it was going to work. So that was why it was a bad flu season. You can have bad flu seasons just when the weather's bad. I mean, if it just spring just comes late, you can have a bad flu season because flu is susceptible. It's an RNA enveloped virus, just like coronavirus, susceptible to seasons. So if you have a longer cold period, you're going to have more influenza.
I think we have time for a couple more here. What's your professional opinion on the extent of the stay-at-home order, whether it's going to be extended past May, or what you might think are the next steps for Ohio?
I think everything's going to depend on testing. It seems like Governor DeWine has got a good handle on things. If we can roll out testing, I've been pretty good so far. I told my friends we went caving on, it was March 4th. I'm like, they're gonna shut us down in the next week or so, and I think it was like 10 days later. So I've been pretty good on my estimates. I estimate that it's going to be early to mid-May when the social distancing starts to be released. If the coronavirus is seasonal, I imagine that and we're going to be about testing. If they can get testing online, I think we can be back to finding normalcy in June. If it's seasonal and we get testing online, I would be shocked if we're not. We may have band gatherings above, 100 people or something. I don't know what that might look like, but I think that we'll start to be able to go to restaurants and, walk in the park, return to work. We might be seeing that mid-May to late June. So I think I've been putting bets on mid-May.
I just got to follow up. So with testing, is the main it's mostly a can we ramp up what we have issue. So it's really a business and industry issue. Is that correct?
I think it's got to be government. It's got to be local government. We just got to get enough testing online. Yeah, I mean, you're talking about something like 10 million tests a week.
Yeah.
And I think right now we're still. People are still struggling to get tests. I think we're still in the tens of thousands a week. And that's just got to be, it's got to be at the government level. There's lots of ways you can test. These serology tests would be great because you can test people who don't have the virus anymore. You can just test that they've been exposed. And in fact, I just signed up for a study and there's randomly screening 10,000 people who've not had any symptoms just to see how many have antibodies against the virus to see what the percentage could be. So there's going to be lots of different ways of doing it. It's just going to be scaling, and we have the technology and we have the tools, and certainly Korea has been able to do it. Korea has been doing it since I think February, and they've not had to shut the country down because they find one person and then they immediately shut everything down for that one person. And they've even been doing cell phone tracking. And I know it's like everybody's like, oh my God, my privacy and everything. It's like, well, what do you want to happen? But like, they've been tracking cell phone data. So if people like pass each other in the park, they know that that's a track and they've got to find that person and they got to shut them down too. And that's how you keep it going.
I don't know what our timetable here is, Hazel. Is it 30? I don't want to keep.
I'm getting a bit tired now, so maybe we'll do one more and then we'll call it a night.
Well, let me finish off the last one, which is an important follow-up is, so if you think it's going to wind down around there, is it going to affect the football season?
It all depends on season and when we get a vaccine. So if we get, I know Tony's been saying that we'll get a vaccine within 18 months. We've never taken anything to clinical trials as quickly as we have done now. They could be rolling out vaccines in the fall. If we can get a vaccine online, it's a scaling issue as well, like these RNA component vaccines. I don't know if you've ever synthesized RNA, but you buy some from the IDT DNA technologies and it costs you like, it's like $10. It's not much. And you get like 100 nanomole. So that's probably a quarter of a grain of sugar. And that's what they synthesize per run on their machine. And we would need over 250 kilograms to be able to vaccinate everybody in the US. So it's a scaling issue. It's going to need new technologies and new approaches. There are some self-assembling vaccines, which seems like they might have promising approaches. And that might be something where the vaccine actually makes copies of itself and self-assemble. It sounds kind of like Star Trekky and everything, but it's a really clever idea and it might work great. So there's going to have to be some technology advancements. The other thing is antivirals. We've got a lot of treatments. One of the things that they've been finding is that patients who have really severe forms of this disease actually have disseminated coagulation. So they get blood clots all over their bodies, and those blood clots can actually end up in their lungs. So now they're treating as if somebody was having a stroke. And so that's increasing, you know, the likelihood of people recovering. So there's lots of things, like it's literally every day we know something. And so it's hard to project into the future. A lot is going to depend on testing, seasonality, and then, you know, a vaccine is what we need. Now, if this strain in Asia turns out to be, you know, work as well as we think, and there's a low infectious dose, then, you know, we could have that thing going in months. So there's lots of things. There's all those unknowns, but we know a lot. We're really, I mean, of all the viruses, I mean, I could probably rattle off a dozen now that would just horrify people. And it's like, invest in microbiology. We've been saying about these things for years, but we're in pretty good shape. We could have been in better shape, but we're not in terrible shape.
Well, that's a good place to stop and let you rest your voice. I don't know if there's a way of thanking people over this. The chat's certainly saying a lot of good things, but I'll just say thank you very much. It was a really informative presentation. I learned a whole bunch.
Thanks, Randy. And thanks to everybody for showing up. I know it's late, and, you know, it's kind of like the potential for a really boring lecture about virology. But yeah, you can find me at the University of Akron, Hazel Barton. Just do a Google and we'll get this up on the web. And then if anyone has any questions, just go ahead and email me. We have a lot of students in the program that have taken the course, and we have courses available as well. So anyway, and it's fun. Microbiology is fun, even the scary side.
Thank you very much.
All right, thanks, Randy.
Bye, everybody.
It's good.